Hello all. Today I'll be discussing about virology and a topic on virology that is introduction to viruses. So, despite their simplicity, viruses are extremely important and deserve close attention. Many human viral diseases are known and more are discovered every year as described by the appearance of SARS and avian influenza virus. The study of viruses has contributed significantly to the disciplines of microbiology. Thus, virology has become a significant part of microbiology as well. Viruses have had enormous impact on humans and other organisms, yet very little was known about their nature until fairly recent. Viruses are a unique group of infectious agents whose distinctiveness resides in their simple acellular organization and pattern of reproduction. A complete virus particle or virion consists of one or more molecules of DNA or RNA enclosed in a coat of protein. Some viruses have additional layers that can be complex and contain carbohydrates, lipids, and additional proteins. Viruses can exist in two phases, extracellular and intracellular. Virions, the extracellular phase, possess, if any, enzymes and cannot reproduce independent of living cells. In the intracellular phase, viruses exist primarily as replicating nucleic acid that induce host metabolism to synthesize virion components. Eventually, complete virus particle or virions are released. So, as a whole, viruses differ in from living cells in at least three ways, that is, their simple acellular organization, the presence of either DNA or RNA but not both in almost all virions, and their inability to reproduce independent of cells and carry out cell division as prokaryotes and eukaryotes do. In this slide, this is a generalized structure of a virus. The simplest virus is a naked virus that is basically a nucleocapsid consisting of a geometric capsid assembled around a nucleic acid strand. The next image shows you an envelope virus which is composed of a nucleic acid surrounded by a flexible membrane called an envelope. The envelope usually has viral proteins called spikes inserted into it. Virions range in size from about 10 to 400 nanometer in diameter. The smallest viruses are a little larger than ribosomes, whereas the pox viruses, which include vaccinia virus, are about the same size as the smallest bacteria and can be seen in the light microscope. But most viruses, however, are too small to be visible in the light microscope and must be viewed with a scanning and transmission electron microscope. All virions, even if they possess other constituents, are constructed around a nucleocapsid core. That is, whereas some of the viruses consist only of a nucleocapsid. The nucleocapsid is composed of a nucleic acid, either DNA or RNA, held within a protein coat called the capsid, which protects the viral genetic material, aids in its transfer between host cell. So what basically are capsids is, they are large macromolecular structure that self-assemble from many copies of one or more types of proteins. The proteins used to build the capsids are known as protomers. So the most important advantage of this design strategy is that information stored in viral genetic material is used with maximum efficiency. For example, in case of a tobacco mosaic virus, the capsid is constructed using a single type of protomer that is around 158 amino acid in length. So out of the 6,000 nucleotide that is present in the genetic material of the TMV genome, only about 474 nucleotides are required to code proteins. So this over here is an image. The first image shows an electron micrograph of negatively stained helical capsid tobacco mosaic virus. The second is the image of TMB structure that has a nucleocapsid composed of helical array of protomers with the RNA spiraling on the inside. And the third image shows a proper model of tobacco mosaic virus. So the various morphological types of viruses primarily result from the combination of a particular type of capsid symmetry with the presence or absence of an envelope, which is a lipid layer external to the nucleocapsid. There are three types of capsid symmetry, that is helical, icosahedral, and complex. The virions which have an envelope are called as envelope virus, whereas those lacking an envelope is called as a naked virus. Viruses are exceptionally flexible with respect to the nature of their genomes. They employ all possible four types of nucleic acids, that is, single stranded DNA, double stranded DNA, single stranded RNA, and double stranded RNA. All four types are found in animal viruses. Most plant viruses have single stranded RNA genomes, and most bacterial viruses contain double stranded DNA. So, this table basically summarizes the different variations seen in viral nucleic acids. The size of viral genetic material also varies greatly. Next, we'll talk about segmented viruses. So, basically, segmented viruses are viruses with genomes which are divided into two or more physically separate molecules of nucleic acid, all of which are then packaged into a single virus particle, like in case of influenza virus. Whereas there are viruses with multipartite genomes, means the genomes are segmented, but each genome is packaged into a separate virus particle, which is seen in case of a bromosaic virus. These discrete particles are structurally similar, contain the same component proteins, but they often differ in size depending on the length of the genome segment packaged. Talking about the classification of viruses, in around 1971, International Committee for Taxonomy of Viruses developed a system, that is ICTV system, which described almost 2000 virus species and placed them in orders, 3 orders, 73 families, 9 subfamilies, and 287 genera. They which place greatest weight on specific properties to define the families, that is nucleic acid type, whether the virus is a DNA virus or RNA virus, nucleic acid strandedness, that is whether it is a double stranded virus or a single stranded virus, the sense of single stranded RNA genomes, whether it is a positive strand or a negative sense strand, presence or absence of an envelope, that is whether it is an enveloped virus or a naked virus, symmetry of the capsid, that is whether it is helical, icosahedral, or a complex symmetry, and dimensions of the virion and capsid. Moving on to the nomenclature of viruses, viruses order names usually end in virales. Viruses family names usually end in viridae, subfamily names virinae, and genus and species name usually end with virus. So here's an example of Mononega virales, which usually has 4 families, the one of which is Paramyxoviridae, which usually contains 2 subfamilies, one of which is Paramyxovirinae, which consists of 6 genera, out of which 2 of them are rubella virus and morbillivirus. Rubella virus has an example under it, that is mumps virus, and that of morbillivirus is measles virus. Thank you.